All right, take two. Um, I actually recorded the whole thing, and I realized I didn't hit the word record. So, yeah. Anyhow, let's do this. Okay, for you guys the first time, for me the second time. All right. From the current slide. All right, protein synthesis and secretion. Okay, so protein synthesis is also called translation. Okay, in the previous thing, we went from DNA to RNA. Now we're going from RNA to protein. Okay, this is translation because we're literally translating, okay, from the language of nucleotides to the language of amino acids. Okay. Okay. So you take the mRNA and you look. Okay. You attach it to a ribosome, and not only do you attach it to one, you attach it to many ribosomes. Okay, like they go in order. One attaches, starts, and another one attaches, starts. So it's not like you go through it once and then twice. You go through it once, and as soon as there's room, you attach another one on. As soon as there's room, you attach another one on. And that's why you make protein so fast. Okay, so this is a polyribosome. Okay, and so here, okay, you can look at the RNA. The RNA is split into three letters at a time. Those three letters at a time is called a codon. Okay, what the ribosome actually looks for is the very first methionine, very first AUG. Okay, that is the start codon. So you search the RNA for the very first AUG, and that's where you start. Okay, there are three letters, right? So first, second, third position. Each of them can be A, G, C, or U. Okay, so 4 to the third power is 64. So there's 64 different combinations. Okay, each of the combinations either codes for the start codon, which is there's only one, and then there's 3 stop codons, and then all the other ones code for different amino acids. Okay, most amino acids have more than one codon. Okay, for reasons why I really don't want to explain. All right, so what you do is you take the RNA, you take the DNA, and you look for the messenger RNA. Okay, so this is the DNA template strand. Okay, the other strand is called the coding strand. Okay, so the template strand binds to the messenger RNA. Okay, so and looks for the first AUG. Okay, and then goes to codon number 2 and looks for whatever that is, matches that up, matches codon 3, 4, 5, 6, 7, all the way until you hit the stop codon. Okay, or run into a stop codon. At a stop codon, you stop. Okay, at each position, okay, at each codon, you have a tRNA. So this is what tRNA looks like. Okay, it looks like a T, roughly, but it's called transfer RNA. Okay, the tRNA attaches, and at the end of the tRNA is a site that attaches to amino acid. Okay, so it matches the codon to the anticodon, and once that matches, that puts the amino acid onto the new chain. Okay, and then everything moves over one, and you put on a new tRNA and a new amino acid, and you move over one, and then you put on a new tRNA and new amino acid, and you move over one, all the way until you hit the stop codon. Okay, once you're at the stop codon, the whole entire thing falls apart. Okay, All right. So that, okay, these are the 3 sites on the RNA or on the ribosome. They're the EPA sites. Okay, you don't need to know that, but you know that they move over one. Okay, and you put on a new nucleus, you put on a new amino acid, and then you move over, and it's like a little assembly line. You chug along, right, 3 bases at a time. Okay. Okay, so the stuff that's being made, okay, has 2 possible fates. The proteins that are being made have 2 possible fates. One is inside the cell. Okay, inside the cell can mean inside the mitochondria, inside the cytoplasm, etc., etc. Okay, the other one is that it gets exported either to the cell membrane or beyond, so outside the cell. Okay. For those that have to go outside the cell, okay, it has to go through the rough ER. Okay. All right. So here's how it goes. Okay. Once I start, so the yellow is the protein. Okay. The blue is the RNA. And so you first spit out the first 30 bases are going to be this thing called a leader sequence. The leader sequence is extremely hydrophobic. So being extremely hydrophobic is extremely uncomfortable here. Where it's comfortable is embedded in the membrane. Right in the middle of the membrane is hydrophobic. Okay, so now the head of it is put in here. The leader sequence ultimately is cut off. But the head of it is already in, and so the rest of it just follows, okay? And you either get something sticking out of the end of the membrane, or you get something that's completely inside the membrane, okay? Okay. Okay. From there, they go to the Golgi apparatus next. Okay? So all the stuff that goes into the rough ER will eventually be taken to the Golgi. The Golgi functions like a post office. Okay? It packages it, tags it, labels it, like this to the cell membrane, this to the outside. It literally tags, puts little tags on there, okay? And puts little sugars on there to display it, okay? Then they're shipped to their destination, okay? So just like post office, tagging and shipping. Okay, proteins, once they're made, are right on the clock. You do not want proteins floating around forever. Okay, that's very dangerous because what you're trying to do 1 minute is not what you're trying to do the next minute. So once you make it, you need to set a timer on it and give it a chance to die. Okay? So where proteins will die is the lysosome. Okay? So I gotta figure out a way to put a timer on the protein, and once that timer goes off, it has to go to the lysosome. The timer is ubiquitin, okay? Once the protein is made, ubiquitin starts attaching itself to the protein, okay? Ubiquitin is small, so a small amount of ubiquitin can attach to it doesn't make a difference. But as more and more and more ubiquitin gets attached to it, it starts to not function and starts to go to the lysosome. The lysosome picks up the ubiquitin and takes it into the lysosome. Okay, so as you build up more and more ubiquitin, it goes to the lysosome. Okay, all right. That makes sense, hopefully. Okay. Okay, I'm gonna go through this rather quickly. I know this is difficult though, so if you have questions, just send me a send me a message, and I'll have a one-on-one chat with you, and that'll make it easier to explain. Okay. So. Okay. DNA synthesis and cell division. Okay. Before cell division, okay, the DNA has to replicate because you want the DNA replication to occur safely, and then you divide the cell in half, and half of the DNA can go to each cell. Okay. The phase that the DNA replicates is called the S phase. Okay. Okay. It involves two special types of enzymes. One is a helicase. Okay. Helicases break hydrogen bonds between the DNA strands. Okay. So, let me show you. Okay. DNA. One strand, other strand. Okay. You'll notice very quickly I can't draw. Okay. But here in the middle is just hydrogen bonds, right? I showed you that earlier. A helicase comes down the middle and separates the two chains. Okay. It comes down this way. Okay. Okay. So the way we usually talk about it is unzipping. Okay. It literally unzips the DNA. Okay. There's also a bunch of dirty jokes about helicase and getting into pets of people, but I won't repeat any of those, but you can imagine them. Okay. All right. All right. Once they're unzipped, okay, you can add to both sides of the ladder now. Okay. So if I have A and T, okay, so if I have A and a T, this side where the A is, I can add a new T. This side where the T is, I can add a new A. And therefore, this is called semi-conservative replication. Because this side has the old A and the new T, and this side has the new T and the old A. Okay, so semi-conservative. Half of it is old and half of it is new. Okay. Oh, and the enzyme that does this is called DNA polymerase, right? Makes new polymers of DNA. Okay. Okay, cell cycle. Okay. The cell cycle is divided into interphase, mitosis, and cytokinesis. Okay, so when we first understood the cell or start looking at cells, what we understood was literally nothing. And then we started cell divide, and we started to see the stages where the cell divide, and we understood that rather well, and we call that mitosis. But because we didn't have enough really good molecular biology material, we didn't know what was happening to the rest of the cell, other than the fact that was the mitosis, because that part was rather visible. Okay, so what we called it was interphase, in between mitotic phases. Okay, eventually what we figured out was the S phase. Okay, the S phase here was DNA replication. And once we could label DNA, we could see DNA replication, and therefore we knew what that was, and we called it the S phase. Okay, for DNA synthesis. Okay, the S is for synthesis. Okay, so that left this gap on the right and this gap on the left. So G1 is actually gap phase 1. G2 is gap phase 2. Okay, they have to be retrocon, so you can you hear the terms growth phase 1 and growth phase 2, but the real name for it is gap phase 1 and gap phase 2. Okay. In growth phase 1, okay, the centrioles replicate. That's one of the things. But the most important thing is one cell is divided in two here. So these two cells are a little bit half the size of that one. So it needs to grow. Okay? So most of the time, cells are in G1. Like they're just growing, getting bigger, doing their thing. Okay? At the very end of G1, the centrioles actually replicate. Okay? Okay, so there are cells that also don't divide anymore. There are cells that, you know, they're in the cell cycle, but they're not, they're exiting the cell cycle. So they can exit right here. There's an alternative pathway called G0. Okay? Cells in G0 will never replicate again. Okay? Like a lot of your neurons and stuff are in G0 because they'll never replicate again. Okay? And what they do is they turn off all the cellular machinery that's used for replication. Okay? Because you don't want to divide unless you're supposed to divide. So if I'm never going to divide again, then I'm not, you know, I'm not going to waste my time on that, right? Like if I'm never going to drive, I'm not going to have a car. That's not useful. Okay? Okay. So once it goes through, okay, it goes to DNA replication. Okay. There's something here, okay? There is actually a stop sign there, okay? A checkpoint, okay? That checkpoint is manned by an enzyme called P53. Okay, so this is DNA replication. Before you replicate your DNA, you better well check to make sure your DNA is not damaged in any meaningful way. Okay? Or else you'll have errors on replication. And if errors on replication, that leads to cancer. Okay? So P53 actually sits on top of the DNA and actually checks it for damage. Okay? If there is damage, okay? If there is damage to P53, or sorry, if there is damage to the DNA, the P53 has two choices. One, it makes the cell, like if the damage is slight, it makes the cell stop at the checkpoint, repair all the damage, then move forward. If the damage is too extensive, it drives the cell to something called apoptosis or cellular suicide. Okay, there's more on that later, and I'll get to that later, but that's what I want to make sure you guys understand. Okay, all right. Now, once you get here, once you are finished DNA replication, you go to growth phase 2. Growth phase 2 is you finally have one final burst of growth, but more importantly, okay, your DNA is all wrapped up. Right, I explained that earlier. The DNA is all wrapped up in chromosomes. Okay, so if it's wrapped up and blocked, then I can't make any proteins. So I have to make all my proteins and all my enzymes that will get me all the way through before I even enter here. That's G2. Okay, it gets everything ready for mitosis. Okay. So then I use this slide to explain a bunch of things. Okay. 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 There we go. So interphase, blah, blah. Okay. I forgot the part about cyclings. Cyclings, okay, cycle. Okay. So at the very end of mitosis, I purposely destroy all the cyclings. So what a new cell will have no cyclings. As it progresses, it starts to build up cyclings. Okay, when the cycling load gets heavy enough, big enough, it starts the cell cycle again. Okay, overactivity of cyclin D has been implicated in some cancers because overactivity of cyclins, okay, will mean the cell is going through cellular replication faster than normal, and secondly, most importantly, without all the checks and balances built in. Okay. Okay. So I just explained the. This was the G0 thing. This is the P53. Okay. Okay. This is a next chromosome. Okay. Okay. Your DNA is not like this most of the time. I know that, you know, in high school and possibly other biology classes, you know, this is a chromosome. They tell you your DNA is in chromosomes. This is a connect chromosome. This is only during mitosis. Okay, so I'll come back to this in a second. Let me finish all this stuff. Okay, so cell death. Okay, cells have two ways to die: necrosis, which is a bad way; apoptosis, which is a good way. Okay, necrosis is cell dies through some type of pathology. Okay, virus infected or just lack of oxygen, buildup of waste. Okay, cell dies abnormally. That's not a good thing because that means all this, whatever's causing the cells to die is still there. Okay, apoptosis is programmed cell death, and it's performed by enzymes called caspases. Okay, remember from the CRISPR, caspase 9. Caspase 9 is a caspase. Okay, caspases chop up things. Okay, so when you turn on apoptosis, you turn on all these different caspases, and they chop up the DNA, they chop up the bits of the cell. So no matter what's inside the cell, it's destroyed. Like you're purposely destroying yourself. So you leave nothing behind, right? You don't leave any evidence behind for anybody to find. You destroy everything. Okay, there are two ways to do this. One is extrinsic. Extrinsic means that you receive a cell death signal from somewhere else. Okay, a good example is T cells telling you to kill yourself because you're infected by virus. Okay, intrinsic is things like P53. Okay, telling you to kill yourself because there's too much DNA damage. Okay, so not much of P53 removed are used to study cancer and apoptosis because you remove P53, you're much more likely to get cancer. Okay, well over 50% of all cancers have a mutated version of P53. Okay. Okay. So the cell cycle: prophase, metaphase, anaphase, telophase. Okay. Prophase, the first step. Okay. So if I'm gonna take one cell and split the DNA in two, the DNA has to split in two very carefully. Each cell has to get the same DNA. So prophase, what's gonna happen is the chromosomes become visible, okay? So now they're packaged, okay? They go from chromatin to a chromosome. So, sorry, you can't see my hands. They go from chromatin to a chromosome. It becomes packaged, okay? That's the only time you can really see the DNA under a microscope, okay? Because now they're tightly packed together, okay? The second thing that occurs in prophase is that the nucleus disappears, okay? So here you see a normal cell. Here you see there's no more nucleus. See the dark ring there? That's no longer there. The nucleus disappears because you don't need the nucleus anymore. Like the nucleus is protecting DNA, but I need to separate the DNA to opposite sides now. So I need to get rid of the nucleus. The nucleus is now just in the way. Okay? Metaphase is the easiest one to see, and that the chromosomes are all in the middle. Okay? Anaphase, the chromosomes are moving to opposite sides. Okay? Telophase, you're forming a new nucleus. Okay? And you're unraveling the DNA. In a lot of ways, telophase is the opposite of prophase. Okay? So one of the things I like to explain is, like, this is the same thing you would do if you're moving from one house to another. Okay? If I was moving from one house to another, I would need to pack up all my stuff. That's prophase. I pack up all the DNA, okay? And then at the end, telophase, okay, I unpack, right? Okay, cytokinesis, okay? Cytokinesis is splitting of the cytoplasm, okay? You generate something called cleavage pearl. Cytokinesis works the same way like a drawstring bag does, okay? You start off with a big circle around the cell, you pull the string, the circle gets smaller, 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 until it finally just collapses on itself and pitches off. Okay? Okay. So cells can only divide by so long. Okay, so normal cells can only divide a certain number of times. That I think that number is in the 60s. It's called the Hayflick limit. Okay, and that's because every time the cell divides, it loses a little bit of the end of the chromosome. The copying process works perfectly until the very end. You can't copy the very end. Okay, and so the very end of the cells get around this problem by using an enzyme called telomerase. Telomerase makes a telomere. A telomere is just a DNA sequence that's repeated over and over and over again. So you take a DNA sequence, you repeat it over and over again, you put it at the end of the cell, or at the end of the chromosome. Every time you cut a little bit into it, but it doesn't matter because it doesn't mean anything, just like useless DNA, okay? So every time you replicate, you cut a little telomere off. Every time you replicate, you cut a little telomere off. Usually that doesn't matter, but if you keep cutting, you will cut into real stuff, okay? And you'll damage the cell and eventually kill it, okay? So cells that divide indefinitely, such as those in bone marrow, have an enzyme called telomerase, okay, that replicate the telomere. Cancer cells also divide indefinitely, so they also activate telomerase that replicates the telomere, okay? All right. I don't know why they repeat that same thing. Hypertrophy and hyperplasia, okay? Trophy is growth, okay, of size. Okay, hypertrophy is growth of size. Hyperplasia more cells, right? Okay, faster growing cells. Okay. All right. So I'm probably not the first one to tell you this, but at some point in your life, or at some point before you were born, your parents had sex, and your mother contributed half your DNA, or your father contributed half your DNA. Okay. So what they contribute is 23 pairs of, or 23 chromosomes each, and you get 23 pairs of chromosomes put together, or 46. Okay. 22 pairs are autosomes, which means they're almost exactly the same. I'll get to the differences in a little bit. And 1 pair are sex chromosomes. Okay? Each pair is called homologous chromosomes. So you have 1 chromosome 1 from your father, you have 1 chromosome 1 from your mother, you have 1 chromosome 2 from your father, you have 1 chromosome 2 from your mother. Okay? On each chromosome, they'll have the same genes, but not the same alleles. Okay, they can have different versions of a gene, but they'll have the same gene. Like for example, they'll have the gene for eye color on chromosome 1, but that gene can be either blonde or not blonde, blue or brown or hazel, etc., etc. Okay. Okay, meiosis is the process by which 2 cell division steps produce the gametes, the ova and the sperm. This only occurs in ovaries in females and testes in males. Okay? So other than replicating the next generation, meiosis doesn't play a role in the growth and development. Okay? All right, so this is what I mean by homologous chromosomes. Okay, so 2 copies of chromosome 1, 2 copies of chromosome 2, all the way through, and you can see the only thing that's not duplicate of each other are the X and Y. So this is the male. Okay, so X, Y is the male. Okay. All right. The first phase of meiosis. So mitosis is prophase, metaphase, anaphase, telophase. Meiosis has 2 phases. Okay, two stages, meiosis 1 and meiosis 2. Each one is prophase, metaphase, anaphase, telophase. So meiosis 1 is prophase 1, metaphase 1, anaphase 1, telophase 1. Okay? Okay. Meiosis 1 is called the reduction division because you're reducing the total number of chromosomes. Okay? Okay. In prophase 1, this is prophase 1. Okay? There's this really important thing called crossing over. Okay? The 2 matching chromosomes line up and they literally stack on top of one another and they swap pieces of DNA. Okay? Why is that important? Because now every single chromosome is a mixture of your father and your mother. Okay? So if I was to take one of you and look at one particular chromosome, that one particular chromosome will have bits of your mother on it and bits of your father on it. Okay? You don't inherit the whole chromosome from a parent. You inherit parts of a chromosome from a parent. Okay? That leads to an incredible number of offspring that you can make. In fact, every single human in history is going to be different than every other human in history, with the exception of identical twins. Okay. Okay. So, in metaphase 1, the homologous chromosomes line up in the cellular cell. Instead of lining them all up in one row, you form pairs. The homologous chromosomes line up next to each other. Okay, so the 2 chromosome 1s line up. 2 chromosome 2 line up, and 2 chromosome 3s line up, and then you separate them. Okay, that's the reduction. Okay, okay. Now, well, this is a whole crossing over and shuffling chromosomes in metaphase 1 results in genetic recombination and genetic diversity. This is the whole reason why we have sex. Okay? Basically, the reason the crossing over and shuffling chromosomes leads to new combinations of chromosomes that we have never seen before. And therefore, all of us have a new combination of genes. And therefore, in case bad things happen, like, virus wipes out the world, some of us will be able to survive. Okay? Bacteria can duplicate much faster, but when there's a problem, they have more problems adapting. So this is better when that results in more genetic recombination and more diversity, and therefore more possibility that we can survive something. Okay. Okay. So you can see the meiosis here. Okay. Okay. And here you can read the stage of meiosis. Okay. Okay. One of the coolest things I learned in biology, you have the DNA from your mother, you have the DNA from your father. What you didn't know was the DNA from your mother and the DNA from your father actually fight each other for control of you. Okay? And so they try to silence each other, so only their version is expressed. Okay? These silence genes are passed on to daughter cells in mitosis and meiosis without change in DNA base sequence. We usually wipe the epigenetic inheritance clean every time we replicate or every time we reproduce, but not always. It's kind of cool. Okay. All right. Hopefully, you guys understood all that. Okay.